Hi, I'm Corey Casper. I'm the Chief Executive Officer of the Infectious Disease Research Institute in Seattle, Washington, and I'm a Professor of Medicine and Global Health at the University of Washington, also here in Seattle. Today, what I'd like to tell you about are technologies that we're developing here for accessible, durable, and broadly protective vaccines against coronavirus, but technologies that are also applicable to other pandemic infections in the future. By way of background, I'm a physician scientist trained in infectious disease. I've been working on viral infections, the prevention and treatment of these for over 25 years. I'm excited to present to you these technologies today that I think really could be transformative in ending this pandemic. So by way of agenda, I'll first start by talking about the current state of vaccines for COVID. I'll talk a little bit about the gaps that exist right now in existing COVID-19 vaccines as I see them. And then I'll talk about some of the exciting technologies that we are developing here to address these vaccine gaps. And specifically, I want to talk about technology that we're developing to make vaccines stable at room temperature for many, many months, which will help to improve access. Technologies that will lead to more durable vaccine responses. Technology that will allow for a broad protection against multiple viral strains as they evolve or mutate. And finally, talk about something that is not quite a vaccine, but a novel therapy that we're working on here in the treatment of COVID-19. So with all that, let's get into our presentation today. What I want to start with is our view of the current COVID-19 vaccine landscape. And so, as many people probably know, there have been close to 270 vaccines for COVID that have been pursued or developed by researchers around the world. That funnels down pretty significantly when you go from those that are in preclinical studies to those that are in approved or emergency approved indications. When you look at the list, though, of those that are actually available to be put into people's arms as of today, you get the list that is here. And that list that you see is of essentially 7 different vaccine candidates. They're summarized here, and I won't go into all of them into any specific details because that's beyond the purview of this presentation today. But what I would like to address is where I see some of the gaps in these vaccines. So right now, there's a couple of things that I've highlighted in this table that show where I think we still have some miles to go in terms of our ideal vaccine for COVID-19 and for future pandemics. First, you can see the 2 RNA vaccines. These are really some of the lead vaccine candidates here in the United States. They include the Pfizer and the Moderna vaccines. These are mRNA vaccines that are delivered in a butterball or a fat particle known as a lipid nanoparticle. These vaccines have a 2-dose schedule, typically several weeks apart, and they are distinguished from each other by certainly the Pfizer vaccine being needed to be stored at deep cold temperatures, minus 20 degrees Celsius. But they're also distinguished by the fact that they're both close to 95% effective. On the other hand, there are other technologies apart from RNA that are also in vaccines that you can get around the world. Most notably here in the United States are the Johnson & Johnson vaccines. These vaccines are based on a human adenovirus platform, and they're notable for single-dose regimens, the need for less deep cold, so just simple refrigeration as for storage, and an efficacy that may not be as high as the RNA vaccines, but certainly an efficacy that would be enviable for a seasonal flu vaccine. The rest of the list you can see is rounded out by candidates which are much more widely available in the rest of the world. These include vaccines that are based on adenovirus platforms and inactivated viruses. The distinguishing characteristics of these vaccines are that many have multiple dose regimens, many of them are required to have deep cold storage, and at least 2 of them have efficacy numbers which are lower than has been reported, certainly for the RNA vaccines that we're using around the world. So taken together, where I think we still have some gaps are in 3 key areas. These 3 key areas are in access, durability, and breadth. In terms of access, as I've mentioned in the previous slide, the requirement to keep vaccines deeply cold and even at refrigerated temperatures put them out of reach for many people around the world. Here in the United States, we often think that that doesn't apply to us, that keeping a vaccine cold shouldn't be an issue. But I'll show you in subsequent slides, it is an issue here in the United States, and I believe will continue to be, as well as in places where resources are extremely limited and the cold chain is very fragile. On top of that, we have issues of durability. So I'll show you some data later in the presentation to show you how long we model the current RNA vaccines will protect individuals. And there are limits to that. Right now, the current guidance from the federal government in the United States is that the durability of protection is likely about 6 months. Could be longer, but that's right now what we're willing to say. That could be a problem for a virus that shows many signs of becoming endemic. Finally, we have issues with breadth. So really, anyone who's watching the news today is constantly seeing messages about these viral variants, which may reduce the efficacy of some of the vaccines that are currently being used. And so we really need vaccines that will address all three of these issues: access, durability, and breadth. And the good news is that I think we have technology now that can do just that. So going into a little bit more detail about access. So there have been significant challenges to access here in the United States, and that has been most represented by what we're calling vaccine wastage. So what we know is that there's close to 100,000 doses of COVID vaccines that have been wasted within the first several months of rollout here in the United States, despite the fact that most people in this country still cannot access a COVID vaccine if they want it. Here on the right, you can see some of the press clippings, but as you know, when a vaccine is brought to a vaccination center, and we have many of these mass vaccination centers that have sprung up around the United States, when a vaccine is brought to those centers, it must be used immediately. When it is thawed, it can't be restored. And so what that means is that there's this rush to use these vaccines that have been brought to the vaccination centers, and if individuals don't show up for their vaccines, it means that those doses get wasted. So here in the United States, that's been a significant problem, and one that's not getting a lot of attention because we don't routinely track the number of vaccines that are wasted or have to be discarded. Around the rest of the world, though, this is an even greater problem. So in one study that was done a few years ago, it was estimated that 72% of clinics in Africa have unreliable access to electricity. Half of freeze-dried and 25% of liquid vaccines, these are just have to be stored under simple refrigeration, are wasted due to challenges in maintaining this cold chain. And deep cold chains are really infeasible in regions where billions of people on this globe live. There's some photographs on the right. On the top right, you'll see measles vaccines being delivered on coolers here on the backs of bikes in the Democratic Republic of Congo, and you can see that that is a real challenge. And the slide below that, the picture below that is a medicine freezer in Nairobi, a picture that I took a few years ago, showing that even within these freezers, there are often challenges due to the constant electricity surges in maintaining these cold temperatures and with the frosting in the fridges. So really, we need a solution that will bring a vaccine that is stable at room temperature to get the billions of people on this planet protected against SARS-CoV-2. There are important things that we need to think about in terms of durability. So in terms of durability, we don't know the current duration of protection. The current recommendations, as I said, are that these vaccines may be protective for 6 months. But what we do know, and I'll show you some of these data later, are that that the levels of antibody titers, especially neutralizing antibodies, which we think are probably one of the better correlates of protection, to many of our vaccines wane. And so we know that they exceed levels of convalescent plasma for 3 months, but beyond that, we're not sure. So based on some of the modeling data I'll show you later, we have reasons to be concerned that the current vaccines that we have will not be durable, certainly for more than a year. And beyond that, may require constant boosting or annual revaccination. And given the challenges that we've had, even just with a single dose rollout, it would be much more ideal if we had a COVID vaccine that could be durable for decades. Going to the next slide, we also, these are data that, as I mentioned, suggest that that the SARS-CoV-2 could become, rather than a seasonal respiratory virus, an endemic one. So we've already seen the virus persist over 2 respiratory virus seasons. And again, there are many experts who think that these pandemic coronaviruses will circulate for long periods of time. The other reason that I think we need to be concerned is that we know that the level of antibodies that we need to remain protected against these viral variants is higher, and that if antibody titers wane from when you first become vaccinated, those waning titers may expose you to greater risk from the viral variants than even to the wild type, and so the vaccine could be even less durable against variants should they emerge. The final important feature that we'll talk about today that I think represents an unmet need in current vaccines are the breadth of protection. So I just mentioned the viral variants, and the good news is that data suggests that all of the current vaccines that are available in the United States retain efficacy against the coronaviruses that are of different strains from the ones that we're seeing here, whether it's the UK, the South African, Brazilian variants. And so what you can see here is that on the slide on the right, that these vaccines do retain some efficacy against the viral variants, but that it is reduced. And that, again, does the combination of reduced efficacy against the viral variants plus waning antibody titers with the time since vaccination, do those two factors together leave people particularly vulnerable as the epidemic persists and progresses? So with all of this, this hasn't been bright, cheery news. And so rather what I'd like to do is now turn to what we think are tractable solutions that could exist today and be useful for protecting individuals against COVID-19 and its the severe effects of that clinical disease. So the first technology that I'll talk about is our thermostable RNA vaccines, and the second technology that I'll talk about are our vaccine adjuvants. So let me start with our thermostable RNA vaccine. So this is a table that I put together that addresses what I think are the gaps in current RNA vaccines, many of which I've already spoken about, but this goes into it in more detail. And more than that, how IDRI's RNA vaccines address these current limitations. So first, on the left, we've talked about storage and distribution and the need for the deep cold chain for at least one of the 2 RNA vaccines. So what we'll talk about today is that our RNA vaccines are delivered with a novel delivery vehicle that we call a nanostructured lipid carrier. This nanostructured lipid carrier allows the vaccine to be lyophilized or freeze-dried, and in so doing, it allows it to have up to 1 year of stability at room temperature, which we think is a major advance. Next, in terms of potency, our RNA platform is what we call self-amplifying and self-adjuvanting. These 2 factors mean that we get antibody titers after the first dose that are higher than after a multi-dose prime boost regimen. So we think that the potency of these vaccines may also be greater. With that greater potency, we think we'll have a greater duration of immunity because it will take longer for the antibody titers to fall. And finally, we will have a very adaptable platform. And the reason for that is that our platform has advantages in terms of how it's manufactured that make it much more suitable to viral variants. And in that, I'll tell you my story about jelly donuts. So with that, on the next slide, we see the basic design of our RNA platform is an alpha virus RNA replicon based on the Venezuelan equine encephalitis virus. And what we do is we knock out the structural proteins from that alpha virus and replace them with the genes that we're interested in to put into our vaccine. This alpha virus is delivered into the cell effectively by our nanostructured lipid carrier. Once it's in the cell, in the cytoplasm, it undergoes a limited number of cycles of replication. So this is different than mRNA vaccines. And in those 10 or 12 cycles of replication that the virus goes through, it amplifies the target proteins. At the same time, the nanostructured lipid carrier contains an adjuvant, and that adjuvant leads to more potent vaccine responses. So this is novel technology, but what's even equally as novel is the way we deliver it. So I want to tell you about my tale of two donuts. I find when I talk about the story this way, most people love and know donuts, and so this really resonates with folks. So the current vaccines, the Moderna and the Pfizer-BioNTech vaccines, are delivered by this lipid nanoparticle, and to me that is like a jelly donut. So what you have is your flavor of the donut, in this case a raspberry donut. The raspberry represents the strain of the virus you're trying to protect against. So the raspberry in this example is the wild type version of SARS-CoV-2, and the donut is the fat ball, the lipid nanoparticle that delivers it to the cell. What this means is that every time you make this vaccine, you have to put the RNA that you're interested in inside the fat ball, the lipid nanoparticle, and manufacture it in toto each time you want to have a vaccine ready. On the other hand, ours you can think of more like a glazed donut that has sprinkles on it. So in our case, we can make billions of glazed donuts, keep them in simple storage and stockpile them. And then each time there is a new viral variant or a new pandemic, we simply change the sprinkles. So instead of rainbow sprinkles, we may use chocolate sprinkles or we may use vanilla sprinkles. And that gives us incredible flexibility to not have to remanufacture the entire vaccine platform each time there's a new viral variant or each time there's a new strain. It also means that we can vaccinate in a multivalent fashion, much like our seasonal influenza vaccine contains 3 or 4 different strains that are circulating. We can decorate 3 or 4 different strains onto the surface of our donut and protect against multiple coronavirus strains. So in more technical terms, the comparison between the two delivery vehicles is shown here. On the left, you see that the RNA is shoved inside the lipid nanoparticle. On the right, you see our nanostructured lipid carriers. Our NLCs have enhanced stability, and they're, as I'll show you on the next slide, are delivered more efficiently into the cell. They have the RNA decorated on the outside, and yet even though it's on the outside, it remains stable at room temperature for a year. It's attracted to this butterball by electrostatic charges, so the RNA is negatively charged and the phosphate groups are positively charged. And this means that we can stockpile the nanostructured lipid carrier, quickly manufacture the RNA to suit, and this means that we're both scalable and widely ready for the next pandemic. And then within that core of this butterball is a lipid core, but that includes squalene, which many of you may know is used currently as a vaccine adjuvant for many different types of vaccines, but is a potent vaccine adjuvant that we think adds potency to this platform. The data, which are pre-published on bioRxives, and you can see them here undergoing review in a peer-reviewed journal, show that really the experiments on the left show that the vaccine construct resists RNA degradation with RNase challenge for up to 8 months at room temperature and up to 2 years at 4 degrees C. And that room temperature is 25 degrees Celsius. And the graph that's next to in the middle here, it shows the biologic activity of the vaccine, looking at protein expression after it has been kept at room temperature or refrigerated temperatures, a simple refrigeration for 21 months, room temperature on the counter for 8 months. So it really retains activity, just like the freshly made vaccine. And then one of the things that we think is adding to the potency is on the graph on the right, you can see that generally we get about 1 log higher production of protein when we deliver the same RNA via the nanostructured lipid carrier compared to the lipid nanoparticle. So we think we have a potent and thermostable delivery vehicle, which we pair together with our RNA replicon, and together we think that this is a very important solution to the RNA vaccine problem and getting RNA vaccines to every corner of the globe, to every person who needs one. This technology right now has completed most of its preclinical studies. It's working its way through toxicology studies, and we hope to start a phase 1 clinical trial by Q4 of this year. I want to turn now to our potent vaccine adjuvants. By way of background, many people may know that adjuvants are molecules that have been designed to interact with the innate immune system, and their purpose is to increase the immune response against an infectious disease or a cancer. We've put adjuvants in vaccines for close to 100 years. We first started putting heavy metals in vaccines at the beginning of the 1900s. We recognized that by doing so, these heavy metals caused an inflammatory response that increased the efficacy of different vaccines. But we're really in the golden age of adjuvants now, and I would say that that's been heralded by the approval of, I would say, one of the first modern adjuvant formulations in 2017. And this is a system that includes a toll-like receptor agonist, a TLR4 agonist known as MPL, along with other excipients and carrier molecules. And so that first was included in the vaccine Shingrix. That took a vaccine for that was previously vaccines for shingles were about 50% effective and got less effective as you got older and more vulnerable, ironically, to shingles. But it took that vaccine and changed it, changed both the construct of the vaccine and the adjuvant that was used. And in adding an adjuvant to a glycoprotein vaccine, that vaccine became close to 98% effective, including in people over the age of 80. So really that heralded a new age of adjuvants. And now we recognize that this combination of a toll-like receptor agonist and different formulations allows for highly potent vaccines. These adjuvants can be freeze-dried and stored at room temperature for long periods of time. And what we know is that they allow for the protection of vulnerable populations through a number of mechanisms. They allow for broader responses, and they allow for more durable responses. And I'll show you some data that's been generated in the context of COVID, and these are data that are sort of hot off the press and now into pre-publication. So first, here's a publication that you can see the reference below, pre-publication again on bioRxiv, that looks at the addition of an IDRI adjuvant called 3M052 alum that's been co-developed with 3M. This adjuvant, when added to a SARS-CoV-2 RBD ferritin nanoparticle, leads to, as you can see here, about a 1 log higher titer of neutralizing antibodies compared to RNA vaccines. And you can see that it does that both to the D614G SARS-CoV-2 variant as well as the B117 variant. On the right, what you can see, though, is very interestingly that the addition of the adjuvant to the protein vaccine leads to broad cross-strain protection. So by adding the adjuvant, you afford protection against SARS-CoV-1, SARS-CoV-2, and 2 different strains of that coronavirus. So we're very encouraged by this, and we think that the addition of a strong toll-like receptor adjuvant, in this case, a toll-like receptor 7/8 agonist, to a very good protein vaccine could actually allow for cross-strain protection. And more than that, we think that this protection will be durable. So this is also data that's been pre-published now, again, referenced below. But what we've compared is the use of a similar adjuvant formulation, this one instead of with alum, but with a stable emulsion, here SE, so 3M052 SE, using that TLR7/8 adjuvant with a protein vaccine leads to significantly higher antibody titers. Both on the left, you can see total IgG, and on the right, you can see pseudovirus neutralization titers, so higher than the RNA vaccines in non-human primates. But also, as you can see on the bottom right, that the duration of protection, and this is antibody titers as expressed by IC50, really the shape of the curve and the decline in the curve as modeled out with the blue line, which is the adjuvanted protein vaccine, affords a significantly longer protection. The time scale on the bottom is on the farthermost right time point is 72 weeks. So again, modeling protection above that lower limit of protection for about a year and a half, and that's we think very impressive for the current state of COVID vaccines. With the last few moments, I want to turn to something that's completely different that we're doing here at IDRI. And so what that is we're also looking at new ways to treat this infection. So we think that there are limitations to current treatments for COVID. While the morbidity has certainly and the mortality of COVID has declined, what we are seeing is that of the treatments that are currently available, we see a couple of limitations. One is that they may not have efficacy across all groups of people who have COVID-19 disease. What I mean by that is that there are several therapies that are most effective in individuals with severe disease or some in very early disease, but few for the full spectrum of patients. At the same time, we worry that the treatment of some of these, using some of these modalities, may render patients more susceptible to future COVID infections by reducing immunity to the virus in the context of treating the acute infection. So we have lots of reasons to think that natural killer cells, which are an important arm of the immune system, play a critical role in viral infections. What we know is that these cells traffic to the lung, that they recognize virally infected cells to become activated, that once they're activated, they kill the target cell, but then they also secrete interferon gamma and TNF alpha that allows the recruitment and priming of T cells. So while they both kill the virus and the virally infected cells, they also may help to establish a more effective cytokine milieu, and in so doing, lead to more effective treatment and future protection against the virus. We currently are leading a phase 1, phase 2 clinical trial of an NK cell product known as Sync01 that is produced by a company in New Jersey called Cellularity. And what we're looking to investigate is whether the treatment with Sync01 leads to better clinical outcomes, as well as a number of translational outcomes in patients with moderate COVID-19 infection. So that trial is currently ongoing. It's a two-phase study with a phase 1 portion that is not randomized to look at safety and tolerability, and a phase 2 portion that is randomized to really look at efficacy. And here you can see some of the data that we'll be presenting over the next several weeks. It's a little too small to really see well here, but of the first 6 participants that have been treated to date, we've seen responses in 5 of those 6 participants. And of those participants, they've cleared their virus. They have had clinical improvements in radiography over a very short time period, as well as an assessment of clinical benefit by their treating clinicians. So we're excited about this therapy. We're excited to complete this ongoing clinical trial, and we hope that this will add to our overall knowledge of how we deal with this pandemic. Most excitingly, these NK cells are off the shelf. They are not specific to COVID-19 and can be very useful for future pandemic respiratory viral infections. So in summary, what I'd like to end with are these conclusions that we've made amazing progress as a scientific community in speeding several COVID vaccines through the clinical development pipeline. I mean, to think of a vaccine going from initial conception to emergency approval or emergency use authorization in just 11 months is nothing short of remarkable. However, we still, the majority of the population around the world and even here in the United States, lack access to these vaccines. And what we need to do is make sure that everyone around the world has access to these technologies and that they have access to technologies that are durable and protect against a broad range of viruses. So with that, we hope that these tools and technologies, our thermostable RNA vaccines, our adjuvanted protein vaccines, and even some of these NK cell therapies may lead to future successes against this and other pandemic viruses. Our institute here in Seattle we're 28 years old. We are a small research institute, but a potent one with a budget of about $25 million in this fiscal year. We have about 65 employees, almost all of whom are in scientific roles. We work on next generation vaccines. We work on global health solutions, as I've just described. And we have incredible manufacturing capacities here in Seattle that serve as a resource for academic, nonprofit, and startup biotech around the world. So with that, I thank you for your time and your attention today. My contact information is here, and I'm always excited and eager to talk to collaborators around the world about the work that we do here and the work that all of you are doing to help end this pandemic and prevent future ones. Thanks for your time today.